Hello. Thanks to meeting organizers for this opportunity to share with you our work about aberrant CTCF binding in the cancer genome using an integrative computational approach. CTCF is a DNA binding protein that can induce DNA looping and maintain the chromatin structure. We created a repertoire of over 600,000 CTCF binding sites in the human genome by integrating over 700 public ChIP-seq datasets and identified constitutive and cell type specific CTCF binding sites. For each cancer, we identified cancer-specific lost and gained CTCF binding sites compared to the global background, shown here using T-cell lymphoblastic leukemia as an example. We found that the lost and gained CTCF bindings are associated with chromatin interaction changes. While most lost sites correlate with increased DNA methylation, many gained sites are not necessarily associated with DNA methylation changes. Using a bioinformatics method we developed, we identified transcription factors bound at regions with increased interaction with the gained CTCF sites, such as Notch in TLL. Perturbation of Notch can lead to binding changes of these gained CTCF, suggesting that gained CTCF are induced by Notch. A knockdown of CTCF leads to decreased expression of Notch target genes, indicating that CTCF is required for Notch to regulate its targets. Putting together, we found that cancer-specific gained CTCF binding are induced by oncogenic transcription factor activities. They function together as enhancers and super-enhancers to facilitate oncogenic transcriptional activation. With that, I would like to thank my postdoc Zhenjia Wang, who did this work, experimental collaborators, and funding support. Thank you very much.